Okay. Hello, everyone. It's a pleasure to be here today. Before I start my talk, I want to say a big congrats to the association for organizing this event, and also thank you for inviting me to present my research and for your financial support, because otherwise I would have not been able to be here today with you. So thank you very much for everything. So the work I'm going to present today has been done in my previous lab in Strasbourg, France, so just a level above Ian's lab. And I tried to put two studies in one presentation, so I hope I was not too greedy. Good thing is, since I'm presenting in late afternoon, I can go pretty fast on the introduction. So you all know the gene we are mentioning today has been identified in Drosophila, proven to be related to neuroblast proliferation, then identified in rats and human. And a few years later, we had more and more mouse and rodent models, some made at the IGPMC. We had overexpression models, we had heterozygous KO models with a lot of different phenotypes and behavior analysis done. Everything to say that, of course, DYRK1A is a gene dose effect. You need 2 copies, you don't need less, you don't need more. Otherwise, you start to have brain development troubles. And so about the syndrome, so the first patient were described in 2008 with 2 big translocations. And then, thanks to the advance of the next generation sequencing technologies, there were more and more mutations that have been identified in more and more patients. So first creation of the association, and so in 2017, this paper actually mentioned that DYRK1A was in the top 10 of the intellectual disability associated genes. So it's still true today. It's a very important gene in the NDD community. So what now? So now I think we are trying to deepen the complexity. Some people work more on the syndrome to refine it, to find more diagnostic methods. This is going to be the first work that I'm going to present today. Some other people work to better understand what is this gene doing, what the protein is doing. There is a lot that has been done. Literature is very large, but there is still to be done here. And finally, there are, of course, some teams that work on inhibitors, but also now on how to activate or having overexpression of this protein. And they actually show some in vivo rescues, which I think is pretty impressive. So sorry if your name is not on this list. It's not exhaustive, so it's not personal. Just before I start to present our work, I wanted to make sure that everybody knows what I'm going to talk about. So basically, if you take a patient, usually a child diagnosed with intellectual disability, he will undergo a DNA sequencing, whole exome, whole genome, whatever. But the end of this analysis is that you end up with hundreds or thousands of mutations, because we are all different. We all carry mutation. And so the whole goal here is first to know if there is a mutation that leads to the disease, because first we don't know what the disease is. And if there is one, the goal is to find which one in 4,000 mutation is responsible for this disease. So I'm not going to go into the details for that, but basically you have several criteria that genetic lab use to classify this variant that we call variant mutations. We look at the genetic informations, we look at the clinical informations, and we can use in silico prediction models, so algorithms that will try to predict what are the consequences of this mutation. So is it like statistically viable to say it's pathogenic or not? And in some very rare cases, we have some animal models that can be, of course, rodent or even flies. And in even more rare cases, we can find some functional tests. So basically what this means is that at the very end, when you compile all this data, you end up with a classification in 5 different classes. Class 1 and 2 that are, the variant is classified as benign or probably benign, which means that you have to look for another mutation. Classes 4 and 5, the variant is probably pathogenic or pathogenic, which means you probably found the diagnostic, so you can give a diagnostic to the family. And in the middle class here, we have the variant called of unknown significance, which means you have clues, you have suspicion that this mutation might lead to a disease, for example, DYRK1A syndrome, but you don't have enough proof to tell the family or to give a final diagnostic. And so this can lead to a diagnostic wandering that can last for years or sometimes decades for families. So our goal was to try to tackle this part and to find ways to classify this variant of significance and be able to say if they were pathogenic or not. So I want to say another thank you to the association for this really nice cartoon that I've been done using several articles in the literature, including ours. So I was honored to see my name on your Instagram post. So basically, as we already mentioned today, you have some recurrent phenotypic traits, in DYRK1A patient, microcephaly, intellectual disability, etc. And when you look at a picture of patient, you will also notice that you can find some recurrent facial features that are not always there, but not in most of the time. And also we have some more variable or rare phenotypic traits that I'm not gonna discuss today. So basically the first goal was to say, okay, so we have these frequent features. What if we try to score them? So if your child has intellectual disability, we give him 2 points. If he has an impaired language, he gets 1 point, etc., etc. And so at the end, are we able to create some kind of a score to predict if the person we see has DYRK1A syndrome or not? So they did this first with a cohort of 23 patients that have been diagnosed to try to establish the score. And you can see that they were all clustering together here very nicely. They did replicate this on another cohort, which worked also very well. And then they tested the score on different pictures of patients affected by other intellectual disability syndromes. And it was working pretty well because you can see that these scores were quite below 10. Some of them were almost intermediate, but it was working pretty well. And of course, at the end, what we did was to try this score with our variant of unknown significance, so patient with mutation in DYRK1A, but for which we didn't know if this mutation were pathogenic or not. And it was quite complicated because some of them were clustering on the top, some of them were clustering on the bottom, but we still have this spectrum here. So the score alone the score is pretty good. It's a good tool, but it's not sensible enough to be used as a diagnostic tool. So that's where I jumped in this project because I'm a bench scientist. So my goal was to model this mutation in cells. So we had 50 variants in this study. I did model 20 of them. You do recognize, I guess, here, so the DYRK1A protein with the 2 NLS sequence leading to the localization of the protein in the nuclei, and also the kinase domain with this phosphate here, which is pretty important. I'm going to mention it after, but basically, DYRK1A need to first phosphorylate this precise phosphate to be able to undergo to its active kinase form. If you don't have this first event of phosphorylation here, your kinase is not going to phosphorylate anything, and we used it later. So we did study 20 variants in total with 4 pathogenic, so mutations that were identified in patients with 100% sure DYRK1A syndrome. We had the wild-type protein and a benign variant, so a mutation that is present in the general population. And we had 15 variants of unknown significance, so mostly missense, 1 amino acid change, and one that was a bit particular at the end, so a distal frame shift that was changing only the end of the protein. So you have about 40 amino acids that were different in this mutant protein. And so the strategy was pretty simple, creating plasmid curve of this variant, transfecting into 2D cells, 3 different cell lines to make sure that we had robust results, and then process to the functional test and compare. So does the protein carrying this variant of unknown significance behave like the wild type, the normal protein or not? The first thing I checked was the cellular localization, because we know that DYRK1A is mostly located in the nucleus of the cell. So you can see here in green that that's the wild type protein that is nicely located. And it was actually the case for 13 variants out of 15. So I will only present 4 variants today, not all the 15. I think it's better for everyone. And for actually 2 of them, we saw a difference, and especially this one, this distal frame shift, we saw that the protein was not going to the nuclei and also aggregating inside the cells. So doing all these dots inside, so protein is basically stuck together. And I did generate a stop mutant here to remove this additional 40 amino acid, and we could see that we were rescuing this phenotype. So this protein is able to go to the nuclei. So it's really the addition of this 40 amino acid that was leading to this phenotype. I didn't check for the protein expression level. So again, transfection, basic Western blot, you have here DYRK1A's protein with the wild type and the 4 variant of significance. For the 15 of them, we could see that 8 of them were actually downregulated. We had less expression. We had a slight decrease for one of them. This one, about 80% of the protein was here. And we could confirm that this decrease of expression was due to a loss of stability. So basically, the protein is degraded by the cell because the cell is able to recognize that it's not going to do its work. So it just goes to the trash and it's degraded. And another test that I used, so coming back on this amino acid, so I told you the protein need to first autophosphorylate in this tyrosine 321, so then it go to the active form. And so I perform immunoprecipitation. You have here, again, the protein, the wild type, and the 4 mutants. And so this is an antibody recognizing the total protein. And then I use another antibody recognizing only the phosphorylated form. And so that way, in the ratio, we were able to show that some of these variants were actually expressed, but they were not able to do this phosphorylation. So they should not have any kinase activity after that. So it was the case, again, for 8 of the 15 variants. And I use, so it's good that Jan presented this too. I use another small assay to check for the kinase activity. So we know the protein tau is a target of DYRK1A. So what I did is I transfected also with tau protein, same amount of tau every time, and a decreased amount of DYRK1A. And we can see when you look at tau phosphorylation that if you have less DYRK1A, you have, of course, less tau phosphorylation. So I did also this assay for all the variants to check for their kinase activity. And for the one that was shown a partial autophosphorylation, we could show that whatever amount of mutated DYRK1A you add in the cell, there is never any phosphorylation here compared to the wild type where you can see the decrease. So partial autophosphorylation doesn't mean you have partial kinase activity. We had the chance to make a really great collaboration with Dr. Rosanna Williamsberg and Eric Chatterjee that are based in Toronto. And so these people are specialists of DNA methylation. So basically, they will sequence the blood, look for methylation of the DNA, and they try to find epigenetic signatures that are specific of different syndromes. So we did send first 10 blood samples from DYRK1A patients, 100% DYRK1A patients, with 24 controls. And here you have the results. So one line is 1 CpG, so one place in the genome where you had a methylation. If the line is yellow, it means that it's over-methylated compared to control. If it's blue, it's under-methylated. And you have, so here you have the DYRK1A. You can see that compared to the control, about 1/3 of the CpG are over-methylated and the other 2/3 are under-methylated. So basically, this means that when you look at DNA methylation, there is a very specific and robust signature of DYRK1A patients. We validate the signature by sending another 6 samples that were all DYRK1A patients. And of course, we sent the blood of the patient that were carrying variant of significance. So you can see that some of these variant of significance were clustering with the control and some other with the patients. So this is just another representation of what I just presented. It's an ACP, so you have here all the controls clustering together. You have here all the patients. So in purple, the discovery of the signature, in red, the validation of the signature. And in gold, you have a bit of everywhere the variant of significance. And we could see that we had 100% correlation between the conclusion we had at the bench, so my functional test in 2D cells, and the methylation signature, meaning that the signature is pretty robust. And so interestingly, we had some like particular cases, I would say. So this threonine 588. So this girl was diagnosed with DYRK1A syndrome first because she has a very evocative clinic. She had all the symptoms on the patients, but I didn't find anything in my cells. And the methylation signature actually showed that it's clustering with the control. So this girl doesn't have DYRK1A syndrome. She's suffering from another condition. Also, Jan did the model of the mice. I put your name here. And they could see that they had also no phenotype for this mice with this mutation. We had another interesting variant here, you may have noticed. It's not clustering with the patient, but neither with the control, and it's a bit more at the opposites of the patient. So this variant that we published in 2019 is suspected to be a gain-of-function variant. Even though I didn't see any difference in my assay, the methylation signature suggests that it might be also a potential gain-of-function variant. So this is a variant that remained variant of equal significance today. So just to give you a summary about this first part, so I hope I convinced you that you need several methods for the diagnostic, and only one is never enough. We found a very sensitive and specific epigenetic signature, and if some of you are looking for potential readout, I'm thinking about KLAN maybe, this could be a pretty easy tool to use because it's already done it. It's about that, you can see, so we reclassified 14 of us out of the 15 we had, so 10 that were pathogenic. So we were able to give 10 diagnostic to 10 families, which is, I think, the greatest achievement of my PhD. And we have here also one remaining variant of significance as a potential gain-of-function. So there are some limits, of course. There is a paper that mentioned that depending of the ethnicity of the patient, the score might not be super relevant because depending of the ethnicity, you don't have the same facial features. And so what about feature variants? So we would advise you to use a blood sampling and do methylation analysis. Find the autophosphorylation if you want to do it in vitro. And if you have a low clinical score or an absence of methylation signature, then go for the kinase activity. And so I'm having a call here because so in this lab there is a PhD student that took this project over. She's now working on investigating more and more gain-of-function variants. So if you're interested in this or you identify mutation in very early or very late position of the protein, please reach them out. Just so second part of the talk, I'm going to switch to so the better understanding of the implication in cellular processes. So I think we will have this kind of slides too, just to say that DYRK1A is basically doing everything. I was able to add this immunity box recently. And concerning the interactors, so there is a lot of interactors that are published more and more each year. We are about more than 1,000 interactors today. And again, this is something we saw today. So DYRK1A is involved in, of course, Courraud syndrome, Down syndrome, but also now a lot of different pathology from cancer, neurodegenerative, and everything. So what was bothering us at that time is that when you look at omics concerning the DYRK1A in the literature, that can be transcriptomics or proteomics, there's a lot of different studies made in different tissue with different cells and in different animals models. So we had ELA cells, we had zebrafish, for example, or xenopus for sequencing data. And it was actually kind of the same with the interactors, so different models and everything. So since we were working in the brain and there was at that time no human tissue, no endogenous studies either, because usually it's either overexpression or siRNA. We wanted to add our contribution by using a human model. So we did use human neural stem cells, which are able to self-renew. That can be also differentiated into neurons. So cortical neurons, most of the population you get are neurons and a mix of astrocytes and oligodendrocytes. And so these cells, we had two lines. One had been generated from embryonic stem cells, and the other one was generated from induced pluripotent stem cells. So the idea was pretty simple. It was to give an idea of the landscape. So what is this protein doing in these cells? What are its interactors? So we did proceed to transcriptomic interactomics using mass spectrometry. And we checked out the proliferation of these cells when we inactivate DYRK1A. So concerning the interactors, we used two different antibodies. So one recognizing, the C-terminal of the protein, and the other one recognizing the N-terminal of the protein. And so we did immunoprecipitate and use this extract in mass spectrometry. We identified 35 interactors, 20 that were not published at the time. Maybe it's the case now. Very few phosphorylation events, surprisingly. So, of course, the tyrosine 321 DYRK1A and 3 FAM proteins. And we had enrichment in especially cell cycle protein, ubiquitination, and ECM protein. Among these 35 interactors, 16 are associated with other entities with other neurodevelopmental disorders, which might suggest that there is maybe not a common path, but there are some pathways that might be connected between entities. And 11 has moderate to high positive correlation with the DYRK1A expression during brain development. So we also went for transcriptomic. We used siRNA to inactivate DYRK1A, partially inactivate. So we had about 40 to 50% DYRK1A remaining. We found about 90 differently expressed genes after the sequencing. A lot of them of this protein were involved in extracellular matrix. A lot of them were downregulated. We had only 6 that were upregulated. And as I told you, we had 2 different cell lines, so I did use qPCR to test this gene, and I was able to confirm manually that 23 of these genes were also downregulated or upregulated in the other cell line. Always good to have different cell line to validate the results. Among these genes, interestingly, we had 2 transcription factors or activating genes, so E2F and EDR receptor pathways. And we could find also that the target genes of this transcription factor were also upregulated or downregulated in our sequencing. Interestingly, we found a specific isoform of this P21 protein that was upregulated in our siRNA condition. And so I did validate this using qPCR. So we have here the control cells and the cells inactivated for DYRK1A with the 2 different cell lines. And very surprisingly, when I looked at the protein, so the P21 protein or CDKN1A, it's the same protein. I could see that when you inactivate DYRK1A in both cell lines, in fact have a decrease of the protein, which is quite surprising because we see an increase of mRNA. So it could be kind of a compensation by the cells. You have less protein, so the cells try to produce more mRNA to counter this decrease of protein, but this is something I can prove today, but this is one of my hypotheses. And so P21 is a very important protein, especially involved in cell cycle and cell division. So that's why we checked what was happening on the proliferation of the cells. So you have here in green and in orange the controls. So the cells are dividing on 4 days. So we were basically counting the number of cells every day from day 0 to day 4. So control condition, you have more cells. That's what you expect. PLK1 is a positive control. So if it's working, if the experiment is working, it's going to kill the cell. So that's why you have no cell after PLK1 treatment. And when you inactivate DYRK1A, we could see that we had a huge decrease in cell proliferation, which was not that a surprise because it has been shown in mice, flies, and a lot of different models. And so when we came back on the mass spectrometry results, we saw that we had this particular protein, so RNF114, which is ubiquitin ligase, that regulate P21 levels in the cells. And if you remember, we had decreased P21 levels. So I first validated the interaction between DYRK1A and this RNF protein using immunoprecipitation. And then I could show again that, so we had less P21, sorry, when we inactivate DYRK1A. And I also made another experiment where I inactivate this RNF protein, and we had the same result. We had less P21 also, which was, again, also here surprising because we were expecting to see an increase. It's ubiquitin ligase. So one of the final experiments was to try to see if we had a link between these two. So I made another proliferation assay where this time I had two negative controls here. And I also inactivated the cells for this RNF protein together with DYRK1A, I mean, alone or together, so both of them inactivated. And we didn't see any difference of proliferation for either RNF 114 alone or any difference when you inactivate both of them. So meaning that DYRK1A, of course, decreases the proliferation, but the link between the two is not sufficiently long to impact the cell cycle. One of the hypotheses that we could have is about P21. So P21 is a protein that is very low or not expressed in adult in the brain. And we can see when we differentiate our cells into neurons, we can see that P21 expression decrease across time. So this is true at the mRNA level, this is true at the protein level. So maybe, and this is in accordance with what has been said previously this afternoon, we have here a decrease of proliferation because the cells are prematurely entering into the differentiation process. So summary of my talk, I gave you a few speech about the variant, the functional tests, the DNA methylation signature that we identified, and a potential gain of function variant that is now ongoing. We also had this description. It's not more than this, but I think it's good to have. So what is happening in human neural stem cells? What are the genes that are regulated, misregulated when you inactivate DYRK1A? What are the interactors of DYRK1A in this precise cell type? The effect that reduces the cell's proliferation, and so potentially between DYRK1A, RNF, and P21. And right now, so the lab is keep going on this. So now it's not really time of siRNA into the cells. So the lab has jumped into a brain organoids model with iPSCs that carry endogenous mutation of patients. And so it's too bad that the next speaker is not here today. But anyway, it is what it is. So yeah, final thanks to all my previous team, especially Amélie Piton, which is still leading the project. So she's the person you should get in contact with if you are interested. Solen that took all the project and is still working on it. All the collaborators and of course patients and families, because there is no research if you are not participating too. So thank you very much also for this. And I'm happy to take any question. It's the end of the day, so I get it. It's fine.
I'm wondering, so what is your opinion about the epigenetic? Very specific for us.
Yeah.
But you said it could be that or it could be used by people if we are able now to reactivate the DYRK1A.
It's a very good question. I don't have the answer, obviously. So what we know is that we didn't found any specific gene that were affected by this mutilation. This is more like spread mutilation over the genome. I think it would be first very interesting to do the epigenome in cell cultures to see if we can replicate this in the dish. And if it's working, then why not use it, as a biomarker? Because epigenetic is very dynamic, so I would be surprised if there was absolutely no changes if you are able to like rescue DYRK1A. Yeah.
Sounds pretty easy earlier. I've got a lot of questions. The one thing I had was, did you buy any chance to look at P53? You know, P53 that each one play on some different DYRK1A is known as far as
I know, but we didn't. So yeah, it was not in the interactors. We didn't detect it either in the RNA sequencing. But yeah, this is something that I should suggest to Emily, definitely. Yeah.
One thing I know that, I think from your work, it establishes that understanding the functional consequences of the mutation is so important. And I think that's Chinese sample versus a data, it behaves a little differently in terms of like mutation or what. And I think it's very difficult to get the mutation and say, okay, yeah, this is the mutation. It's either likely pathogenic, or it could be a variant of significance. But really, like the next step from there, I think, is a gap oftentimes, because for families who just have the information, it's up to you almost to connect with researchers to say, okay, well, like how much is it kind of suspected, or is this? Because I think a lot of times the assumption is like it's either a friendship or nonsense, and you're just not producing anything. So I think just for our community, it still emphasizes the importance of just understanding what is that next step beyond the mutation, because I think that's where it becomes really important to understanding how you address it. And
I'm curious about what was found in the patients that you discovered that they didn't have the Courraud syndrome. Is there certain additional syndromes, or is there just different groups of diseases that we might have some different subgroups in the future?
So far we don't know. There was another very deep sequencing that has been done to try to look for something else, or a mutation that we would have missed with, let's say, more classic sequencing, and they didn't find anything. The thing is also, we should not forget that sometimes you have environmental causes for this kind of syndromes, which means that you can look at the DNA, you won't find anything. So this is one of the limits, and one of the problems is when you find nothing, it's when do you stop looking for it? Because if there is nothing to find, you will never find it. So yeah. Thank you.